wanted to go to the next
Yeah. I remembered it when I was putting the lecture together. Thank you. Okay, guys. So we don't have too much to talk about today because somehow we're ahead of schedule, which is good. Which means two things. One, we can spend a little time today going over ways for you to organize the information to make it easier to study. And two, Monday we can have a review session. So I think the goal here will be finish the last, it's only 9 slides that I have. Actually, probably 7 when you count that this is a slide that doesn't have content on it. So the goal would be finish those, go over some ways to organize the information, because I've had a lot of students visit me and everyone's like, I don't know how to put this all together in a way that I'll remember it. So we're going to try to work through that today, and then you can spend, this afternoon through the weekend organizing yourself and thinking the way I'm telling you to think. And then on Monday, come in with specific questions, because people are coming to my office with very specific questions that are really awesome, and people are walking away with a hopefully satisfied look on their face. Okay, so we'll do that in a minute. So this is my call for exam questions. Again, just like last time, a whole bunch of you got some points. Even 1 point helps, right? So by Monday, please email me a multiple choice exam question clearly about this stuff. Some people emailed me exam questions that had nothing to do with what we had learned. And I just want to say to you, what kind of teacher do you think I am? You think I'm going to test you on something I didn't teach you? That would be so mean. So you guys have some very evil teachers in your class. And make sure it's analytical. Make sure there's no none of the above. Make sure there's no all of the above. Make sure all of the answers are feasible. You know how to do it. And if it's a perfect question, I don't have to do anything, you'll get 4 points. If I need to mess with it, but it gave me a great idea for a question, you'll get 1 point, and it'll be added to your exam grade, so it won't show up in your extra credit thing on Blackboard. Monday, 5 p.m. Okay, and then we'll go over the thing about organizing the information after. So let's just blast through these last things. These last few slides are all review, because it's all about turning off signaling. And we already know. Like, I had someone come visit me today who was like, let's talk about turning off signaling. Would it be this, and this? And I was like, well, that's today's lecture. So, yeah. So hopefully that should take your stress level down a little bit, because you're not being thrown any more real new material. But it's really about bringing it all together. Okay, so here's just some thoughts and concepts that you should be asking yourself while you're studying. What is the purpose of this thing I am trying to remember? If it doesn't fit in with a story and it just seems like it's a fact that's floating in space, then you need to stop yourself and back it up and figure out where it fits in. Because if you're just memorizing pieces, it's going to be hard. And then with everything we talked about, there's regulation. So ask yourself, how is it a regulated process? What is the regulation in this? How is it being turned on? How is it being turned off? Right? Because we all understand signaling is highly regulated, because when it's dysregulated, you get bad things, including cancer. Yes. Why is it regulated? Right. Because you don't want expression of a gene that's going to push you into S phase and mitosis if you don't need to, right? What would be the outcome if it became unregulated, right? So if a G protein suddenly got stuck in the on position, what would happen to the pathway that you're thinking about? How could signaling become unregulated? Many ways, right? Mutations, mostly mutations. But when I say mutation isn't just one thing that turns something off. A mutation, depending on where it is, can affect different things. It can affect a protein that it's interacting with. It can affect, ability to hydrolyze GTP. It could affect ability to interact with a GEF. It could affect so many different things. So you need to really think through these things, okay? Good. Okay, so just things for you to think about. And if you've got no idea what any of this means, email me. Okay, so you saw this before, and I'm bringing it back up because you need to understand that signaling needs to be turned off once the purpose was accomplished, right? Signaling is very purposeful because there's usually a ligand that binds to a receptor. Ooh, misconception that someone brought up to me today that I really would like to take a moment to clarify. Who thinks that receptors bind to ligands and bring them into the cell? No one thinks that? Not one person in this class thinks that? No one thinks that ligands are ever brought into the cell. Oh, God. Okay, we're in trouble. LDL receptor, transferrin receptor, are they bringing their ligands into the cell? Okay, so I want you to acknowledge the fact that sometimes yes, but sometimes no. And there are two ways it could be. Sometimes the ligand, what do I mean by ligand? Does anyone know what a ligand is? Right, it's the signal, it's the red ball. The signaling molecule. So, in some cases, something does need to be brought into the cell, like with the LDL receptor or transferrin receptor, right? Sometimes all it's doing is activating a signaling cascade. And I want you to keep that big picture in your mind that there's not just one way for anything with biology. There's multiple ways. And I thank the person who brought that up today because it's these questions that make me realize that sometimes I'm not very clear. Okay. Okay. So signaling needs to be turned off. How is signaling turned off? You guys already know. This is not a new slide. You've seen it before, but let's just go through it for kicks, right? You'll have once you have your ligand binding receptor, you can have endocytosis, and then it could end up going in a recycling endosome and then back up to the surface later. Or it could go into what other kind of endosome? Now, what kind of endosome? A multivesicular endosome and just be sequestered and then later go back up. It depends on the situation. And then I did hear another one. The endosome could then go to the lysosome, right? So that's this one. So one, you're kind of sequestering, you're hiding your receptor until it's needed again. And the other, you're hiding your receptor and then you're trashing it. So those are two ways. You see how that's done regulation, right? Another, you can have a protein that binds to the cytoplasmic tail. Because what happens on the cytoplasmic tail of a receptor? Signaling. That's where it all happens. If you chop off your cytoplasmic tail, will any protein be able to bind to it? No, because it's not there. Right. And so it's all about phosphorylation and creating docking sites and recruiting other proteins that are going to function in transmitting the message to the nucleus or to affect the cell's function or whatever. And so if you have no cytoplasmic tail or if you've obscured it with a protein that's binding and hiding things, then you're going to be inactivated. That's another way. And you can inactivate the signaling protein. Here it's not the receptor itself, but it's a signaling protein. What's an example of a signaling protein that could be inactivated that will stop transmission of your signal to the nucleus? A G protein is one, right? So that with the GPCR, if you inactivate that, right? That's the whole thing with phosphorylation and arrestin. Well, that's one of the things that happens. Another signaling protein that's really big, you can regulate its activity, is RAS, right? If you inactivate that, then you're not going to have any signaling. Production of an inhibitory protein. Sometimes a protein will be activated that will then have a feedback loop, right, and actually inhibit the receptor. And so we had an example of that also, right? Does anyone know? Oh, I kappa B? What does I kappa B bind to? I love it when people put themselves out there, so thank you. Because it's okay to be wrong. I kappa B binds to NF kappa B. So that's like way down in the cytoplasm somewhere. But you can have an inhibitory, and I kappa B is an inhibitory protein, but it's in a different context. I kappa B, yeah. So one example was, yep.
GSK technically be inhibitory protein, since it phosphorylates beta protein and then makes it.
GSK is an inhibitory protein, but does it inhibit the receptor?
No.
No. Yep. So you're coming up with a lot of really great ways that signaling can be inhibited. But when we talk about inhibiting the receptor, arrestin, right? So go back to arrestin, where you're producing, right? Remember, you got like, so imagine that that's your GPCR. It doesn't look like that, I know. And that's your G protein, right? And you have activation of adenyl cyclase and then cyclic AMP and PKA, and then PKA comes back up here and phosphorylates the tail and recruits arrestin and decouples. Remember, we learned all about that. So that's another example of downregulation. So this is all at the receptor, right? And as a lot of people just brought up, there are other ways of turning off signaling that aren't at the receptor level. But we'll get to that in just a minute. So receptor endocytosis, we learned about this 50 times, so I really don't want to have to go through it too much. But one thing I want you to think about with arrestin that is specific for GPCRs, that doesn't mean only GPCRs are endocytosed. There's another mechanism for that with RTKs, or the enzyme linked receptors. But in terms of arrestins, it binds to the receptor and it recruits clathrin, right? So what if you had to think about proteins that bind to receptors and recruit clathrin, what do you think of? Yep. Adaptin, exactly. These adapter proteins that are involved in vesicle budding. Remember when we learned about vesicle formation and clathrin vesicles going from the Golgi to the lysosome? They're totally different proteins, but have a lot of similarities, and I'm guessing might either share domains or interact with similar proteins. And you could probably look it up and find that. So it's always nice to kind of think back like, oh, I've seen this before. This looks similar. The only difference is here this is on the plasma membrane, and it's happening budding out of the Golgi when you have vesicle formation, right? Okay. So receptors, they could be endososed and recycled. They're sent to the lysosome, and this was the one that we talked about way back when I talked about endocytosis. Now, I think I confused a whole lot of people with talking about endocytosis in conjunction with trafficking of lysosomal proteins to the lysosome, and I apologize. I've learned from you guys that this is where I need to talk about endocytosis. Probably not over there, but this is really what it has to do with, right? Not really anything about sending acid hydrolases to the lysosome. Right, so remember we learned about the LDL receptor, and you get your clathrin-coated head and your vesicle, and then it goes over here, and the pH drops, you get your recycling endosomes, and you get your endosomes that go to the lysosome. Old news, right? Okay, and then we talked about this a fair amount, right, where you can sequester things in multivesicular endosomes. What's the purpose of a multivesicular endosome versus just a plain old endosome? I'm hearing hides the cytoplasmic tail, right, because you can have a signaling endosome if it's not sequestered, right, because the cytoplasmic tail is still exposed to the cytoplasm, right? And we learned about this in terms of beta-catenin and GSK3, all very fair game for the exam. Okay, and you can also have receptor phosphorylation, which is similar to what we learned about with arrestin. And this is just another example where you have a GPCR and a hormone and cyclic AMP. Cyclic AMP increases due to signaling, and that results in phosphorylation of receptors, which then turns off production of cyclic AMP, and also activates. Then you have the phosphodiesterase, which hydrolyzes cyclic AMP, and then you have low levels. But just basically, that's kind of a confusing way to think about it, and I don't know why I put that one back up there. But the Arrestin model is a much more clear way to understand it, right? Because if you phosphorylate a receptor, you're not going to be able to interact with the signaling proteins, right? You can deactivate your G protein, right? G proteins don't stay on. They get turned off. How do G proteins get turned off? Yep. Right, so GTP hydrolysis. And what determines whether GTP is going to be hydrolyzed? A protein with what kind of function? Gap function, right? So these RGS proteins. Or sometimes just binding to the target protein itself will have that gap function and induce GTP hydrolysis. So you want to turn off your G protein after you bind to your target, right? That makes sense because you've activated your target protein. You're making plenty of cyclic AMP. If we keep that G protein on, well, that's not good, right? We want to like transmit our signal, then turn it off. And so this is the last slide. Can you believe it? I just flew through that. This is the last slide, and we started with JAK-STAT, and we're going to end with JAK-STAT, right? So the inhibition of JAK-STAT can happen through, sorry. There's a whole thing that's not in here that I really want to be in here. Okay. It'll be on the board. So let's go back to JAK-STAT over here. So JAK-STAT is inhibited in many different ways, okay? So we already talked about receptor endocytosis, but then another mechanism is through degradation of different components of the signaling pathway. Does that make sense? If you want to turn off a signaling pathway, you can degrade your receptor, logical. Why not also degrade some of your kinases, right? If you degrade all of your JAKs, will you have signaling through JAK-STAT pathway? No, and so that's one of the things that happens is normally you have signaling, and that activates gene expression, and then some of the genes that are induced are involved in degrading JAK to help turn that pathway off. And so that's inhibiting JAK kinase activity, inhibiting receptor phosphorylation, inhibiting the whole signaling pathway. And so one thing, I don't know why it's not here, that I brought up before and I want you to also remember is the role of phosphatases, right? What do phosphatases do? They dephosphorylate. They remove phosphate groups. So if you dephosphorylate your receptor, what are you doing to the proteins that should be involved with signaling? You're causing them to not bind, right? So whenever you think about phosphorylation, and this has come up a number of times, people think about phosphorylation as activating. Phosphorylation is not, you should not think about it as activating. You should think about it as changing the shape of something. So sometimes you're changing the shape of your receptor and creating docking sites for other proteins that will function in signaling. If you take off that phosphorylation, then that docking site is gone and it can't recruit the other proteins. Sometimes phosphorylation is inactivating, right? Sometimes phosphorylation, as we'll learn when we talk about cell cycle, sometimes phosphorylation prevents interaction with things. So definitely don't get caught in that phosphorylation equals activation. Think of it as changing the shape, right? Because protein-protein interactions are all about having surfaces of proteins that can interact with each other through their biochemical properties, right? So if you throw a phosphate group on there, it'll either encourage it or inhibit it, depending on the proteins, okay? Okay, so that's it. So let's go back to, what I started out. How do we organize all of this information, right? Who here feels like they're on it? And who here feels like it's all this information and they have no idea how to approach it? Good, so I'm not wasting my time. So someone came to visit me yesterday and asked that question. I said, well, and we were going through some stuff from some lectures, and I realized he didn't have any hierarchy to his thinking. And so he developed a little bit of hierarchy, and I think that helped a little bit. So, so we've got signaling, right? And under signaling, we start at the top, right? I like to start at the top. We'll start with receptors. No, let's not even. Let's not even. Let's start with ligands. Okay, so what kind of ligands are there in terms of biochemical properties? Hormones. Okay, and are they hydrophilic or hydrophobic? Or do they bind to hydrophilic carrier molecules or hydrophobic carrier molecules? I'm trying to go take you in the direction of, will it pass through the plasma membrane or not? Okay, so you can have ligands that are hydrophilic or hydrophobic in nature, okay? Some are hormones, yes, definitely. Will hydrophilic ligands go through the plasma membrane, just diffuse through the plasma membrane? No. Okay. Will hydrophobic ligands diffuse through the plasma membrane? Yes, right, because the plasma membrane is hydrophobic. Okay, so what kind of receptors do they bind to? Nuclear receptors. Exactly. Okay. So hydrophilic, will they go through the plasma membrane? No. So then they probably need to bind to something on the surface of the cell, right? Because otherwise, how are they going to get their message into the cell, right? So they cannot go through, and there were 3 kinds that we focused on, right? So can't diffuse through plasma membrane, so need to bind cell surface receptors. Okay, so tell me the 3 cell surface receptors you guys are studying. GPCR. Okay. RTK, receptor tyrosine kinase. Enzyme associated. So you can say enzyme associated would be up here, and so let's fix this because they're kind of the same family. So we'll go enzyme coupled, and then RTK, and enzyme associated, or kinase associated. Okay. Is everyone with me so far? Are people starting to see how things are flowing a little bit? Okay. So then what you need to do is think about how does the message get to the nucleus? Okay, and you can start to differentiate there, right? With nuclear receptors, what needs to happen? So you already know you have a hydrophobic signaling molecule that diffuses through the plasma membrane and binds to a nuclear receptor, right? So with nuclear receptors, what happens to allow the nuclear receptor to activate gene expression? Exposes the NLS, maybe in some cases, but even more so, what does it expose? The DNA binding site. So here's where you draw your picture of your nuclear receptor, which has multiple domains. You'll have your hormone binding domain and your like DNA binding domain and your transcription, activation. And it's all about, sorry guys, you can look it up, it's in your notes. And it's all about, who thought a nuclear receptor was on a membrane? It wasn't, right? No. It's a soluble protein, and its activation all depends on hormone binding and inducing a conformational change, which exposes the DNA binding domain. So normally you'll have an inhibitor bound, and then the hormone binds, and inhibitors kicked off, and coactivators are recruited, and there's this DNA binding domain that can bind to the DNA. There's my DNA, and activate transcription. Yes? Does the DNA binding domain sort of act as an NLS? No. So it's a nuclear receptor because it's frequently in the nucleus. It has an NLS somewhere. So that's the thing. Two mechanisms. So you can even go over here. Mechanisms for inducing gene expression really have to do with ability of something to bind to the DNA at the promoter. And how do you control that? You can control two different ways, right? Right? You can have access of the NLS to an important, right? So NLS, and that allows a protein to go into the nucleus and activate gene expression, right? So NLS access, or you can have DNA binding domain access. Maybe they're going to change the lights again. Did someone go out?
Yeah.
I was hoping someone would change the lights again. Okay. Right, so right, because they're both different forms of regulation. It all comes down to, can the transcription factor bind to the DNA? Either the DNA binding domain is always available, and you're regulating it by regulating whether or not your transcription factor is in the cytosol or the nucleus, or your DNA binding domain is often not available, and it could be in the nucleus. Who cares? Because don't forget, your hydrophobic signaling molecule diffuses through the plasma membrane, diffuse. It can go all the way through. You can have some that are in the cytosol, so it's not that it's all, but it really more has to do with this DNA binding domain access. Because whether an NOS is exposed or not, if you don't have your hormone binding, which allows your DNA binding domain to be exposed, you're not going to bind to the DNA anyways. Okay, let's go back to this. So that's nuclear receptors, right? And nuclear receptors is the simplest form of signaling, right? There's no kinases and relaying signals, none of that, right? Because your signaling molecule is doing all the work by diffusing all the way down to the receptor, which is right near the DNA. And it's just a conformational change and binding to a promoter and activating gene expression. Okay, so that's how we activate gene expression through nuclear receptors. How do we activate gene expression through GPCRs? Anyone? So you want to think of it as a sequential process, right? right? So you've got a signaling molecule. Let's start on the outside. Signaling molecule. We already have it, right? Signaling molecule that happens to be hydrophilic binds to the cell surface receptor called the GPCR, which results in activation of what? A receptor. But the receptor through what? Well, the receptor, and then it activates a G protein, right? Now the G protein's active. How is it activated? GDP, the GTP, right? So now your G protein's active. What's the point? What's it doing? Phosphorylating an enzyme. Activating an enzyme. Activating an enzyme. Do G proteins phosphorylate? They're not kinases. Activating an enzyme. Right? So then G protein activates an enzyme. Oops. And what's the example I gave you guys? What enzyme gets activated? No one knows? Adenocyclase. So enzyme, example, and it's a plasma membrane bound enzyme in this case. Example, I'm writing AC for adenocyclase because I'll be there forever. Okay. Adenocyclase, what does it do? Makes cyclic AMP. Cyclic AMP, what does it do? Activates PKA. PKA, what does it do? Goes in the nucleus, activates gene expression. So what you have is basically production of a small molecule, right? So adenocyclase makes a small mediator. Here, let's come back over here. GPCR, right? Continue. So you have adenocyclase activated, or enzyme. Let's keep it. Keep it broad, and then you have some kind of small meteor. A meteor comes, and then then you don't have to take the exam because we've been hit by a large meteor, an asteroid actually. Who wants an asteroid to hit so that we don't have to take the exam on Wednesday? Okay, I like honesty. Okay, so small mediator. Are small mediators proteins? Is cyclic AMP a protein? Oh my God, guys. Nod. I see a nod. Thank you. Thank you. I ask you like the most simple questions, and when no one answers and everyone just stares at me, it makes me feel like you don't know the answer. And that scares me so much because it makes me feel like I've just been standing up here doing like a cha-cha for a few weeks. Okay. Cyclic AMP, GMP, they're cyclic nucleotides, right? Look up the structure. Okay. Small mediators activate some kinase. Gene expression. Activates kinase, and the kinase activates gene expression. Through phosphorylating something, right? Because kinase is phosphorylate. Okay. So we've done nuclear receptors. We've done GP. Do you realize I'm teaching you the whole exam in like 20 minutes? Okay. Okay, enzyme coupled. So RTKs, what's the difference between RTKs and kinase associate? What's the difference? Right, RTKs. Intrinsic. So it has kinase domain, right? Okay, and where does this one get its kinase activity from? Kinase associated. A kinase that it associates with. Come on, people. What color is George Washington's white horse? Purple? Okay. So binds kinase. Okay, so how do these guys activate gene expression? Right? So that's really the only different, right? There. So what are things that are similar with the cytoplasmic tails of these? Yeah, I just saw someone go like that. Cross, right, so they dimerize when the ligand binds. Remember, let's start over here. Hydrophilic ligand binds to the cell surface receptor, and you either have intrinsic kinase activity or kinase that's bound that cross-phosphorylates the receptors, right? So you've got this cross-linking of the receptors. Right, so you'll have phosphorylation. What is phosphorylation of the receptors doing? Creating binding sites, right? So then recruits other proteins. Okay, and so in the simplest form, you have the JAK-STAT pathway. Those other proteins get phosphorylated, and then what do? They dimerize and go to the nucleus, right? That's the simplest. There's other proteins. So then what we could do here is you can go in many different directions because they're all the same to recruit other proteins, and then there are different mechanisms. And I gave you examples, right? I gave you JAK-STAT, I gave you NF-kappa B, I gave you Wnt. What else did I give you? Oh, I gave you Notch, right? So that's where you need to categorize them. Okay, so that's it. Recruits other proteins. Let's go back. Oh dear. Here, I'll make that go up. You want to power it off now? Okay. I just need to make it go away. Where's the no-show? No-show. There. Okay. So now we're in enzyme-coupled receptors, right? How are we doing? We're good. We're good. We're going to make it through the whole exam in like half a class. So the question is, uh-oh, people are getting excited. The question is, enzyme-coupled receptors, yes? Oh. Transcriptional activation domain, yeah. So you'll have your DNA binding domain, but you have to interact with activators that help to recruit RNA polymerase. So, And I'm doing this on 1 cup of coffee. That's it. I just want you to know. So that means you guys will need at least 2 when you guys come in. Okay? Okay. Enzyme coupled. Now we're comparing, contrasting different ways of activating gene expression, right? Because the whole point of the signaling is to relay some message and get something to go into the nucleus and bind to DNA and activate gene expression. How that transcription factor makes it into the nucleus seems to be what differs, right? So there's different ways. So example 1, right, which is JAK-STAT, where you have dimerization of JAK-STAT, right? Of STAT. Dimerize STAT. What does that do? It basically creates an NOS. Goes to the nucleus. Gene expression. Okay, so then we had all these pathways that had to do with degradation of something, remember? So let's make that like a subtopic, right? Degradation. of things, right? So what did we degrade? We do in NF-kappa B. What did we degrade? Inhibitor, right? So the inhibitor, what's it normally doing? How's it inhibiting the transcription factor from going to the nucleus? If you had to guess and you knew nothing. It covers the NLS, so you throw it in the garbage and then you can see the NLS, right? The great inhibitor. Oh, look, the same nucleus. Gene expression, right? What else did we degrade? Give me another thing. What did we degrade? Come on, guys. Yep. Okay, so GSK3 is not degraded, but it is in a pathway with degradation. What's degraded in the Wnt signaling pathway? Beta-catenin, right? So the Wnt, right? So in this case, it's the reverse. Without signaling, you're constantly degrading beta-catenin, and you need to stabilize something, right? So it's the opposite. So when so this is all like we're talking about activating. So to activate, you need to prevent. Yeah. Do you guys want to give me an A in penmanship or an F? Prevent beta-catenin destabilization or degradation. Beta-catenin is a transcription factor. It's always being degraded. You need to not degrade it. So that's the difference, right? When you prevent degradation, then it can go into nucleus. Then the NLS is exposed because the protein's there, right? So there, the NLS is not being hidden. The whole protein's being degraded, right? And then, same thing. The mechanism is different, right? Notch was another one with degradation. Does anyone remember? And it was still different, right? Notch, actually, you have cleavage of the cytoplasmic domain of the receptor, right? So, cytoplasmic domain of receptor cleaved. So it's actually not degradation, it's just cleavage. And then it goes to nucleus and activation expression. Whew! Okay. Did that help, or are you guys more confused?
Yeah.
Okay. So if you start to map things out like this, this is what will make you successful. Those of you who want to go to med school or any of those kind of scary schools where they try to kill you, you might want to really work hard on figuring out how to organize information so you can think about it. So now I've given you a starting point. You have to go through yourself some of the more detailed things, but now you have a framework. And so when you talk about GSK3 and all that, you at least know it has to do with activation and enzyme coupled and Wnt and all that. We have some more time. If anyone has any specific questions. Yes?
So these are all examples of each type of signaling. So I know you said that we need to know that they are just examples, but we need to know like the IKB.
No, you can say, let's say I have, it helps to know it's NF-kappa B, but even if you were like these words, like, like if I had to learn like a foreign language with a different alphabet, like Chinese, it would be really hard for me because these are sounds that I haven't heard before. And so sometimes lots of words can become like alphabet soup. So I get that. So if you were able to say you degrade an inhibitor exposing an NLS and you understood that concept, you'd be golden. Yeah. So however it best works for you. So for me, I remember it by the pathway because I understand the pathways. But for you, if you understand, because what you really need to understand is these are all different mechanisms for preventing or for allowing gene expression. So you've got all this signaling. The signaling all starts and all that, But then really the goal is at the end, what goes into the nucleus, how is that controlled? And so those are all different, right? Any other questions? Yeah, so if I had an essay and I said, give me 5 mechanisms for regulation of gene expression based on signaling or something like that, you would say, well, you could expose the NLS through dimerizing, you could expose the NLS through degrading an inhibitor, you could prevent degradation of something that's normally being degraded, and you could you can release a sequestered transcription factor, which is essentially what is happening with Notch, because it's normally stuck up at the cytoplasm. Okay. Anything else in the last minute or two? Okay, good. Come on Monday prepared with questions. I don't want to stand. I will not do this again. And everyone who didn't come today missed out.